Alright, squad, I got a new piece of paper and I am on page 8 now. Everything that we've already learned at the beginning for the most part. At the bottom, I made a mention about viruses. You don't have to know anything about viruses really at this point. You just know that there are different types of viruses and that, like out there in the world, like with viruses, it's not so simple as like DNA is always linear, or like DNA is always double-stranded. Like there's many different types, and we do not consider viruses as living things. So like generally speaking, if you see like a DNA strand that looks like U A C G C U A, then you would assume that would be RNA. If you see a DNA strand that looks something like A T T G C C T A, then you would assume that that's DNA because of the T's. For the most part. Going on to page number 9. Please attempt this page and then pause for now, attempt the page, and then unpause, and then I'll go over the answers. Hopefully y'all have attempted the answers. And what comprises the backbone of DNA? That's going to be sugar and phosphate. What type of bonds hold together nucleotides? So if you're talking about between nucleotides, then it's going to be hydrogen bonds. If you're talking about within a nucleotide, then that's going to be covalent bonds. Summarize the scientific contributions of Chargaff, Franklin, and Watson and Crick. So Chargaff, that's going to be all about A's and T's going together and G's and C's going together, and then me putting 2 lines in between to represent 2 hydrogen bonds, me putting 3 lines here to represent 3 hydrogen bonds. Then we got Franklin, which discovered the double helix nature with crystalline or crystallography, X-ray crystallography. And then Watson and Crick combined everything to make the full and complete 3D model of DNA. Regarding the practice FRQ, please make sure that you already did the practice FRQ before pressing play on this video and watching this. Please pause if you have not already done it. For A, identify the location of the antibiotic resistant genes. That would be the plasmid. That would be the location, because what we're essentially talking about is that we have a bacteria with its own bacterial DNA, and then another bacteria with its own bacterial DNA. But then one bacteria has this special DNA that can be conferred or transferred to another bacteria. So the traits on this bacteria that make it resistant, these are for resistance. The traits are for resistance. They are going to go from one bacteria to another via the circle being the plasmid. B, and then how so? So, like, how is it getting transmitted, basically? If it goes via the environment, like, so for example, let's say that we destroy this bacteria, all of the bacteria's guts are everywhere, the DNA is all over the place, all the contents are all over the place, the cell membrane is destroyed, everything is just kind of like in the environment. Well, what happened is that the plasmid can go from the environment straight into the bacteria and then give it the plasmid. Just go straight through via the environment. So via environment, just go straight in, or via direct contact. From one bacteria to the next. So let's say that this bacteria is alive, this bacteria is alive, and then they form a bridge, and then the plasmid from one bacteria goes to the other side. This would be the second type, via direct contact, and this structure right here is called a sex pilus. And then there are names for these two that we don't need to know exactly right now, but I'll tell you. Via direct contact, the name for that is going to be, I believe, I don't want to get this right, so I'm going to look it up. I believe it's conjugation. Okay. And then the environment, I'm going to look this up real quick. Just to make sure I spell it right. Be transformation. And then the one with the viruses. If we were using viruses, then that would be transduction. So I'm just going to do it in a different color and be like, side note, if we were using a virus, then that would be, so with virus, that would be transduction. There we go. All right. Moving on to the next page, we are now going to be on page number 10. So I'm going to delineate the notes right here. Say page 10. That's where we are at right now. And we are now going to be talking about 3 different models of DNA replication. The correct model is going to be the semi-conservative model. The other models are proposed. They're like a hypothesis of sorts, the conservative and the dispersive model. We are able to prove that the semi-conservative model is correct, meaning that here, well, you know what? I'm going to use Legos for this, so that it's easier to see and easier to tell. Okay, let's see if I can. Okay, there we go. All right, I got green. Let's see if I can get any more colors in here. We got blue. Yeah, okay. So we have our parental DNA strands. These are going to be the nucleotides. This right here is the parental DNA. We have the left strand and then we have the right strand, or the top and the bottom strand. Either way you want to see it. So these are the parental strands. And then we're going to do DNA replication. We're going to open up the strands. And we're going to add new nucleotides. So one strand is going to be able to create another DNA molecule. One strand is going to be able to create another DNA molecule. And then boom, there we go. let's say that we want to do DNA replication again. So now I'm going to do a new color. Let's do pink. Yeah, let's do pink. Okay. Hold up. Okay, we got our pink. So, if we wanted to do DNA replication again, we're going to do replication on both sides. And these two will come apart. One will go to form another DNA molecule. One will go to form another DNA molecule. These two will come apart. One will form another DNA molecule. One will form another DNA molecule. There you go. And that's how it is semi-conservative. So these right here, the green, these are like the grandparents. And then these are like the parents. And then these are like the grandchildren or the children, so to speak. We are partially conserving, meaning that we are conserving these DNA molecules. They don't just go away. They don't just disappear forever. And they don't stay together. They get reused. Each strand gets reused. Okay. We were able to prove that by using isotopes, nitrogen 15 and nitrogen 14. So let's say that these two right here, we have, I'll write it down too. Let's say that the green, we used nitrogen 15, which is heavy. And let's say that we have pink. That's going to be the new, the new DNA. We culture the rest of it. We have the parents in the N15, and then we culture after the first round of replication. We culture everything else in N14, which is light. So where the N would go, the nitrogen would go into the nitrogenous bases. So if you look at DNA molecule, you'll see that you have the phosphate group, you have the pentose sugar, and then you have these nitrogenous bases, and they're full of N's. They're full of nitrogens. So the culture, the nitrogenous culture, they're going to supply the nitrogens to these bases. So the bases are going to be heavy in the beginning, and then after the first round of replication, they're going to be very light. So the way that this works is that we have the green, which is the heavy, and then after the first round of replication, then we're going to culture the bacteria in N14, which is the light. So this will be the first round, generation 0. This is what generation 1 looks like. And then this is what generation 2 looks like. There you go, generation 2. And as you can see, the way that we have exponentially less and less green, or you could see it as exponentially more and more pink as each generation goes on to the subsequent generation, that proves that we have the semi-conservative model. Okay, next page is going to be a little crazy. That's going to be page 11 over DNA replication. Okay, a lot of stuff going on. And that's why I have the Legos. Alright, I got a bunch. So, let's try to figure this out by using Legos as part of the model. Okay. Alrighty. Okay, there we go. So, let's figure this out. And you know what? This is a lot easier than you think it is. This is not too bad. I wouldn't worry about it. Don't worry about it. It's not as bad as you think it is. So, you have DNA. Boom, there you go. That's your DNA. And nothing's happened yet. Your DNA is just sitting there in the nucleus. But we're going to have to replicate it in preparation for cell division. So what we're going to do is we're going to have to open it. So the way that we're going to open it is with helicase. So let's say that we are going to open it at the end. You can start anywhere. There can be multiple sites where you can start replication, and each site is the origin of replication. There can be multiple origins of replication at the same time. Let's say we're starting at the end. Here we go. Okay, let's say that this is like just a long DNA. We're going to open it at the very end. And what opens the DNA is going to be the helicase. Let's see what we are going to use for the helicase. We're going to use... boom. There we go. So I'm going to write on the side the thing that looks like this. That's the helicase. So this thing, that's the helicase. It's going to keep this open so that it doesn't close up again. Keeps it open. And to keep the DNA from binding to itself, there's proteins called single-stranded binding proteins, or SSBPs. So this unwinds the DNA. It keeps it open, it unwinds it, it goes down like a zipper. I want you to think of the helicase as a zipper. Okay, there we go. And then we have the single-stranded binding proteins that help make sure that the DNA does not get back together. So let's say that this is like the single-stranded binding proteins. There we go. There we go. So this right here, there we go. This is going to be the single-stranded binding proteins. They help keep these two strands from going back together. We have the topoisomerase. And this right here, this thing that looks like a door, this can be the topoisomerase. It goes over here to keep the DNA from winding up too tightly. So I'm going to go over here and say that this door-looking thing is going to be the topoisomerase. And the point is that it keeps the DNA from supercoiling. So prevent supercoiling. There we go. So what supercoiling looks like, I'll show you with a rubber band. All right, we have here rubber band. And the whole point is that, let's say that I am going to be taking this DNA let's say that this is a DNA strand. You can see the helix in the middle. And let's say that I am going to be opening it up more and more on one side. So I'm making one side much bigger than the other side. As you can see, the rubber band is coiling up and getting extremely tight. And because of that, if you coil it more and more and more and just kind of like pull on the DNA and make it tighter and tighter, the rubber band can snap. And we don't want the DNA to snap. So what the topoisomerase does is that it prevents that. It keeps the DNA loose and a double helix like this, and it prevents the DNA from supercoiling, which would happen if you pulled on it too much. So this is the topoisomerase to prevent supercoiling. Then we have the enzyme primase, which initiates the replication by adding short segments of RNA called primers. All right, so here I'm going to extend the DNA. There we go, there we go. Oh, right there and there. So this is the DNA. There's two strands to this DNA. And we are going to finally put down a nucleotide. But we are going to put down an RNA primer first. So let's say that this right here, let's say that this is the primase. The primase puts down the primer. So the thing that looks like this, that's going to be the primase. ACE means enzyme. So this is the enzyme that puts down primers. And a primer, let's say that this little ball is the, well, no. Let's say this one, the yellow one. Let's say this yellow one, this tiny yellow one. Let's say that that is going to be the primer, specifically the RNA primer. It's going to be made out of RNA. And I know you're thinking, this is DNA replication. Why are we putting down RNA first? Well, that's just the way it works. Think of it as you have to put down like a rough draft before you finish the final draft. So the rough draft is going to be the RNA primer, and it's not going to be there like forever. You're going to put in DNA eventually, but you got to put this down first. So we have the RNA primer coming down, being attached by the primase. So let's do it over here. Boom, there you go. Cha-ching! Pretend that sound was the RNA primer working. And then we're going to do another one over here, because again, we're going to do it on both sides. Cha-ching! So notice I put down an RNA primer over here and over here, because DNA is anti-parallel. So the way this works is that this right here, this DNA strand, is going to be the leading strand because it leads right into the fork. And this right here is going to be the lagging strand. It has to wait for the fork to open to put down new nucleotides. So the way that the nucleotides are going to be added is going to be like this. But then this helicase is going to keep opening up the fork. So imagine this goes down. and we are opening it up, right? So we're providing more space. There we go. We're providing more space for nucleotides to drop down. Let's say that the helicase keeps moving downward. There we go. Moving a little bit more down. There we go. And then we're going to open it up again a little bit more. Boom, there we go. Opening up more DNA to be available for replication. As you can see, we got this extra space here, and we got this extra space here. So if we're in the leading strand, we can just go straight into the fork, just like that. No interruptions whatsoever. But here, for the lagging strand, we started over here. We're going to go out, but then, oh no, we have to restart again. So now I'm going to take another yellow piece. We got the primase. Okay, got the primase. We're going to take the primase and we're going to move another primer over here downstream. So then from this point, we can start DNA replication again. Boom, there we go. And then we're going to end up gluing these two pieces together with the ligase. So this again, on the leading strand, DNA replication is continuous. And then on the lagging strand, the DNA replication is non-continuous. Okay. It is the DNA polymerase 3 that attaches each primer. Hold up. It is the DNA polymerase 3 that attaches nucleotides extending each primer. So let's say that the new DNA is blue. So this right here, this is the new DNA. This is the RNA primer. This is the new DNA. This is the old DNA. So this is like the parental DNA. This is the daughter DNA. These are the new strands, the blue. This is going to be added down one nucleotide at a time. Right here, boom, there we go. There we go. One nucleotide at a time. And because it is the leading strand, again, it's continuous, it goes straight into the fork. And let's say that this circle right here is the DNA polymerase 3. So the circle is going to be the DNA polymerase 3, and it's going to be responsible for adding these down, just like that. We're going to add another one, another blue one right there. Boom, there you go. If we are doing the lagging strand, the way it would work is, again, we have the primase adding the primers, and then we have the DNA polymerase 3 extending the primers with the DNA. So this is extending it. The DNA polymerase 3 is extending the DNA from the primers. And these, by the way, these are free-floating nucleotides. These are just floating around. Like, this is all happening in the nucleus. This is just all floating around, like all these blue and yellow pieces. And then another one comes down. Hold up. Okay. And then another one comes down. DNA polymerase 3 comes down, adds another one. Boom, there we go. And then it goes back. Here we are taking the primase. We're adding a primer right there. And then from this point, we're adding our blue pieces. So there we go. There's a blue piece right there. We're adding that with the DNA polymerase 3. And then another blue piece. There we go, another blue piece. And then what happens is that we need to take out the yellow pieces. We want all this to be blue. We need to take out the yellow pieces. So we want it all DNA. We don't want any RNA in this. So what ends up happening is that we cut out the RNA primers and then we replace it with DNA. What takes out the RNA primers is going to be DNA polymerase 1. So DNA polymerase 1, let's say that it's this big square. It's that big square. DNA polymerase 1 comes in with this, the blue DNA. It swaps out the RNA primer with DNA. And then it comes in everywhere there's an RNA primer, bam, swaps it out with DNA. Because again, we're replicating DNA. The primers were just there for like a rough draft. Or like the foundation for the building, basically. And then, boom. Boom, there we go. And then what we're going to do is we're going to have to glue everything together. We have to take the DNA ligase to join the Okazaki fragments together on the lagging strand. These DNA that are like two different strands, because the discontinuous lagging nature of the DNA replication, they're called Okazaki fragments because the person who discovered them, their name was Okazaki. And this will represent the DNA ligase. They're just going to come in and glue together the pieces to make sure that everything is combined. So when you think of the DNA ligase, I want you to think of ligase as glue. You glue everything together. Let's say this keeps going on and on and on and on and on forever until you reach the end of the DNA molecule. What ends up happening is that everything dissociates from each other, and then boom, this becomes its own DNA molecule. There we go. And this becomes its own DNA molecule. There we go. Perfect. DNA is extremely big, so like you wouldn't want to start at the very end though, if this was like your actual DNA. So DNA replication can start at multiple places at the exact same time, and then all the strands would just get glued together with the DNA ligase. And so DNA replication, it's like, it doesn't just like start at one place and then like, it would take hours basically. It would take a very long time for DNA replication to finish. DNA replication happens at like multiple points, and then all the strands together get glued together by the ligase, and then it can happen much more quickly and much more efficiently. All right, and that's the main things. Move on to page 12. It's more describing what I already talked about. Page 13, it shows you at the very bottom how in eukaryotic DNA there can be multiple replication forks or multiple origins of replication. And then in prokaryotic DNA, it's much simpler and it's smaller and it's a circle, so you only really need one origin of replication. All right. So in the next part, we're going to start off with page number 14.